We'll start with the insights into mapping the brain. I'll request Dr. Arshad Khan Sahib to come and make their presentation.
Assalamu alaikum wa rahmatullahi wa barakatuhu. Bismillah ar-Rahman ar-Rahim. To introduce myself, I am working as an assistant professor at the University of Texas at El Paso. The projects that we have right now at the moment are focused on mapping the landscape of the brain, using animal models to be able to understand the human brain and to understand diseases that might originate from circuitry that goes dysfunctional inside the brain. And so I wanted to present just a couple of sort of glimpses of what I do in the laboratory. The goals of my laboratory are threefold. One is to identify feeding and autonomic control circuits. What does autonomic mean? Autonomic is self-governing parts of the body. You cannot commit suicide by holding your breath. Your body will take over. So there are certain circuits and reflexes in the body that are self-governing. And Hazrat Khalifa Basir Rabe has actually, in his book on absolute justice, kindness, and kinship, talked about the governing mechanisms of the body. And that's been a great influence on my own thinking on how this works. And so the autonomic control of circuits, the reflexive control of hunger, and those types of things are of interest to me in my research. The second goal is to identify drug therapies and treatments that might actually be targeting the brain to be able to combat disorders like diabetes and obesity. And third is to manipulate the brain circuits that we discover in animal models to understand how to control appetite. So if we have the urge to eat, and if we feel like we've already eaten a meal, the dessert, the zarda afterwards, after our meal, is not something we nutritionally need, but we crave and want, and we overeat because of that. Why? And I think it's because the circuits that are in the brain have been primed to associate that food with pleasure. And that pleasure of overeating, though God-given and very good, can sometimes be leading to addiction. Now, I don't know if there is an official zarda addiction, but there is definitely many types of small addictions that we all face like this. So basically, this is work that was performed by my PhD advisor in 1993. I'm showing you here on the screen. These are actually sections of the rat brain. And what he had done in 1993 and published is all of the black dots you see on the screen are areas of the brain in sections in animals where he gave an injection of a drug. And that drug produced the numbers that you see in each of those circles are the amount of grams of food that the laboratory rat, given that injection, ate to the drug. And he found that there are key areas of the brain that actually receive drug and produce a lot of overeating. These animals would eat more in one meal after the drug injection than they normally would eat in an entire day. So these types of experiments help identify in the brain, we share the same brain structures, in fact, what parts of the brain are hot spots that control appetite. And so in my own work, influenced by this, I am asking the question, can we actually find below the drug's actions, what are the circuits in the brain anatomically that are actually triggered by this drug? And that's the goal of my laboratory, to find this out. And many other laboratories, I must add, as well. So we have some idea about brain structure, but unfortunately, you might have heard about the types of scanning we can do. We can do fMRI scans and live scans. You sometimes see this in the news. You can see human patients with lights, hot spots in the brain that light up. Unfortunately, the individual cells inside that tissue that light up are unknown to us, and we cannot see them with those types of scans. They're beyond our resolution. So it turns out that here you have a table. It's a simple table of a number of regions of the brain on the left. And then on the top columns, you see a number of brain regions as well. And the names of the regions are not important, but the colors indicate how the left side column is connected to the top column. So any single place of the brain that has a color indicates a strength of connection. Now, what's interesting about this is that you've heard of the human genome, which is all of the genes that we possess in our cells. Well, there is a new science called the science of finding the human connectome. And the connectome is every brain connection connected to another piece of the brain. And what you see on the right is that matrix that I showed you. But unfortunately, that matrix is only that small portion that you see on the left side. And everything that is gray is unknown about the brain. So 90 to 95% of all neural connections in the brain are completely unknown to us. And this is the challenge I'm facing. How can we find out what is the Zarda circuit if I can't find out anything about the rest of the brain? It's like literally going blind inside a black box. So what we're trying to do is create maps of the brain and try to uncover this using standard techniques. What we actually do is we use laboratory animals. This is an example of brain surgery performed in my lab, where we actually anesthetize animals that are kept alive, and we implant, like my professor did many years ago, the same types of studies. We go in with much more precise instruments, and we actually map those hot spots of the brain that produce appetite, and then try to visualize that. So this is a side view of the rat brain, and the cross section that I'm going to show you is actually shown here. This area of the brain, the hypothalamus, is the area that I study. And the hypothalamus is that area that controls hunger and appetite, among many other things. Now notice here that the areas that you see along with the black stripe in the middle there running along are all very well-defined tiny regions of the brain. But just above it is a large strip called the lateral hypothalamic area. And notice it's blank. That's because we don't know what it looks like yet. And we're working on that. My entire PhD for 6.5 years was trying to understand the circuits of that region. And every single outline you see here on this picture has taken over a century to map. And so this is a tiny dot or small area, less than the size of my fingernail, inside the brain of an animal, and we know nothing about it. So this is the complexity that we're dealing with, and this is the type of work. So just to wrap up, I want to show you a little bit of data on some high-resolution mapping. This is what my microscope sees when I look at a picture of a stained rat brain section. You see a bunch of cells. We have now the ability to be able to see this, not just under the view of the microscope in a small way, but kind of like what you see with Google Earth. You can actually take high-resolution views down to the shoelace and zoom out, and you can see New York City, and you can go in and out of that sort of depth. Well, we can do this with microscopy by stitching the images together in a motorized fashion, so you can actually see a whole panoramic view of a brain section. And this is what you're seeing here. It's a slice of the brain showing the hypothalamus. And what we're now doing is using fluorescence imaging to actually map individual cell types in the brain. And these are views that are unpublished, and they haven't yet been seen. And the idea here is that this green that you're seeing here, I actually showed this in a mulaqat with Hazur. And Hazur says, "Ye main kya dekh raha hu? What am I looking at?" And I said, "Hazur, this is actually green cells of the brain pointed here. And they're sending their wires or their tare to various parts of the brain above it to stop the animal from eating." And so Hazrat Sahib then said that this is the circuit in the brain that actually suppresses your appetite. I said, "Yes, Hazur." So Hazur said, "Okay, so or dikhao, what else do you got?" And I said, "Well, Hazur, we have cells in the brain that are actually there to stimulate your appetite." And these are the pictures of the cells. So what we are now finding is now we're looking at these stained together, and you can start to see the appetite circuits, the wires that are green, touching the cells that are blue. And that hot spot that you're seeing there might be the circuit where an appetite suppressing signal in the brain is communicating with an appetite stimulating, saying, "Bas karo zarda aur na khao." It's kind of enough, right? So that's what we're trying to do. And if we can go into higher detail, because we're doing this with high magnified views, we can actually get to seeing many, many panoramic views of the brain. in such a manner that you can actually, like decks of cards almost, with different suits, you can actually see different molecules in the brain and visualize them at this type of resolution. So this is the type of thing, what you're seeing here is individual nerve cells receiving context and communications. All of the staining that you're seeing here is done by students in my laboratory. And we're actually, this is just one small 100 micron width piece of the brain. And this is the forest of complexity. So I'm scared of my own research. I don't know what this means yet. We're working on it. Jazakallah.
Jazakumullah, Dr. Arshad Khan Sahib. I'm sure you haven't found any Zarda-addicted rat yet, so you may need to come to the humans to find those. MashaAllah, fascinating presentation. May Allah guide you to reach your goal and discover for future humanity.